Hi, my name is Dr. Laura Sakura, and I am an assistant professor at the University of Calgary in the Department of Microbiology, Immunology, and Infectious Diseases, and I am the Functional Omics Platform Lead for the IMPACTT Pan-Canadian Microbiome Core. So I'm here today to tell you about an exciting new research resource that we at IMPACTT have produced for the Canadian microbiome community, and it's a free resource, so it will be distributing it to labs for free. So hopefully I have your attention now. And also, we'll be launching what we're calling our microbiome science benchmarking exercises. And so I just want to start by saying that we at IMPACTT, we've really never saw a need or viewed our role as one that needs to dictate how Canadians conduct their microbiome studies. Instead, we want to support what we know is a common and valued goal of quality and reproducible microbiome science. So we see our role then really to help Canadian microbiome researchers capture and control for methodological bias in their research, because we know this is inescapable. It's something that affects all of the work that we do. We also know that many of you are already doing excellent methodological studies, and we just want to help facilitate that and make it a bit easier. Lastly, this field is still evolving quite rapidly, and lots of things change over time. And so we think that we could play a role in congregating that data and sharing that information that informs optimal methodological choices. And lastly, I think particularly in the past couple of years, we've all been challenged, to say the least, to get any science done. There are still some really big questions out there about how we should study the microbiome, questions that really take substantial time and time and resources to answer. And we see an opportunity then for us in the IMPACTT Microbiome Core to help answer some of those big methodological questions that are outstanding. And so hopefully now we might all have a little bit more bandwidth in our lives to come together, work together to advance our field, and really come up with a new understanding of what we need to do to make robust and translatable discoveries. So what we're going to be doing first involves the use of what we're calling the IMPACTT Microbiome Standards. So defined community standards have a really important role in microbiome research, and they've been used traditionally in sequencing applications, and these standards that we've produced are meant to first be used in that same capacity. So I just want to review quickly some of the really critical features of defined community standards and their applications. So again, this is often a phylogenetically diverse, compositionally defined group of microorganisms that are put together usually in an equimolar or equally distributed manner in the standard, so that you have an expected result when you do your sequencing. You have something that's known and expected, and you can see how well your results recapitulate that. So when we do extraction or sample handling, if we start with the cell pellets of these defined communities themselves, we can evaluate how our sample handling procedures may affect the results of the experiment. We may also, because these are phylogenetically diverse microorganisms, they have different types of cell walls, will ensure that those different microorganisms, gram-negative, gram-positive, are all lysed by our extraction. And lastly, we've been using these recently to really evaluate, mock up and evaluate low biomass situations to see if we're recovering everything we expect and optimizing our signal to noise in low biomass applications. In library prep, this phylogenetically diverse community then will have genomes with different GC contents, and that can allow us to assess whether we have primer bias in our amplicon methods, as well as in our shotgun library prep, whether we have fragmentation bias affecting our results. For sequencing itself, many of you are probably aware that the Illumina platform has an anomaly that can happen called barcode hopping. And in this situation, really what's happening is that the forward and reverse reads can accidentally get split into different samples. And when you're using particularly combinatorial dual indexing, this can actually result in a pair of reads being misassigned to the wrong sample. And so this can happen as frequently as 1% of the time using certain Illumina platforms like HiSeq and NovaSeq. So by looking for reads that are unexpected in your community standards, you can really assess what's happening on your run with barcode hopping. And lastly, for bioinformatics, I'll just put one example here, but when it comes to taxonomic assignment, our community standards can help us ensure that our work is, our methods are sensitive and specific. Okay, but there are also some challenges with microbiome standards, applying them routinely. The first is cost. So by our calculations, some of the cell pellets that we use in those early stages of our analyses can cost up to $50 to $75 per aliquot that goes into our experiment. So that can add up if you're routinely using these. Secondly, access. So there are only a couple commercial suppliers right now that we know of. We use Zymo and ATCC. We've actually had some importation barriers with ATCC because their standards are coming from the US, and even the dead cell pellets that they are shipping for these standards, if they contain group B strep or Streptococcus agalactiae, they can't be imported to Canada because it's a fish pathogen. And also we've had more recently some supply chain issues with some of the Zymo taking months to show up. Lastly, there are limited options, and these communities are often unrealistic or simplistic compared to what we're really assessing, particularly with human feces. And while for the applications I outlined previously, some of that is not a limitation really, you know, it still at times would be beneficial if we had something that was really providing the same types of challenge and complexity to the method that we're really seeing in our true experimental samples. So in that light, we are introducing the IMPACTT Microbiome Standard. This is something that we've partnered with Dr. Emma Allen-Vercoe at the University of Guelph to produce. And she has basically developed this protocol for producing these standards, and she very graciously agreed to produce a set of 4,000 standards for the Canadian microbiome community through IMPACTT. And so this standard has a spike-in or a known, compositionally known community of 45 phylogenetically diverse bacteria that are evenly distributed based on cell counts, and we also know that a lot of these strains are novel. So this is coming from her collection. Most of them had not yet been subjected to whole genome sequencing. We're currently doing that right now. So I don't know yet how many of them are truly going to be novel and more challenging to our annotation pipelines, but we will be finding that out shortly. And lastly, the last thing that makes this really unique is that she spikes this known standard into a human fecal matrix. And so you have that sort of natural substance that is providing quite a bit of challenge really to our efforts to lyse and cleanly remove DNA from these human fecal samples and to really assess then the microbiome composition. And if you don't work on feces, we also have this community spiked just into PBS, which is more of a standard carrier. So I'll tell you a little bit more about how we produce these. So we took healthy human fecal samples from several donors, I think about 8, and we collected these samples, processed them fresh in our chamber, anaerobic chamber. We subjected them to deep shotgun metagenomic sequencing. And then from those taxonomic profiles, we selected 3 that had very different profiles of microbes. Based on those profiles, Dr. Allen-Vercoe then suggested the 45 spike-in strains, and these are exotic strains, not because we've never heard of these microorganisms before, but because they differed substantially from what was already in the samples. So we wanted to make sure that we could expect to really distinguish and detect the spiked-in known standards within the more complex fecal matrix and the community therein. So we then aliquot 1 ml of this fecal slurry with the spike-in into our vials, and then we will be distributing this all over Canada. And like I said, we're hoping to make this free for sure the first shipment, we're still assessing what distribution is going to look like, and whether we can come up with a way to keep it completely free, or whether we need to charge for shipping. We'll let you know soon. So this is what the standard community would look like then if it is perfectly evenly distributed in our experiments. This is what it would look like. These are provisional species assignments that are based on short 16S Sanger sequences at this point. So when we get the whole genomes, we'll be revising the species content. The first shipment that we will send to each person who signs up to receive the samples will include 4 of the vials, so 1 of each type, the 3 different feces matrix components, as well as the PBS with the spike in. We're also producing all of the clean non-human reads, assemblies, and mags from the fecal samples that were used for the matrix. We're also producing the whole genome sequences, and we'll share those draft genome assemblies with the community. And lastly, we'll produce reference packages like a 16S reference package, and we'll be running these through all of our methods this summer to provide analysis recommendations. And then for the benchmarking, we all know that no experiment is perfect, and we really can't escape some degree of methodological bias. But by sharing our results with this common standard, we hope that we can update our knowledge of the do's and don'ts, for which we've learned a lot over the last decade, 15 years in microbiome sequencing. And really what it comes down to today is a set of trade-offs, and also newly available options, methodological options, new kits, vendors. And so maybe we can just collectively update our information about what's working and what's minimizing bias. And so what this will look like is that we hope you'll share your reads from your control samples with us. These are the first set of reads that we generated from the control samples. And as you can see, what we got was really a distribution of relative abundances. These are the 17 species that were over 0.1% relative abundance in our sequencing with V3, V4 variable region. And really about half the reads were from the spike-in altogether, and the other half were from the fecal matrix. And the other 20 or so, 20 to 30 species were below 0.1% relative abundance down to about 0.01% relative abundance. So this distribution of abundances is what we got with our first experiment, and we're anxious to see what others are getting with their experiments. And so we'll be asking that you share your reads from your control samples with us. We'll provide a simple online form that you can use to also upload those reads and then provide methodological details about your experiment. This is a sampling of some of the methods that we will be collecting, or some of the data points we'll be collecting. And then we'll aggregate and share anonymized data with the community, not individual lab-based data, so that we can collectively learn a little bit about really what's important in our experimental designs and what's less important. So we hope you guys will participate, and really we're just getting started with benchmarking, because we know that the sequencing, it's been done before. It's never a bad thing to update, but we are really all about getting to causation and translation, and really where that's at these days is multi-omics. And really also considering not just bacteria. We only have bacteria right now in this standard, but we're going to be looking at making a next generation of standards that are multi-kingdom. We're looking at benchmarking metabolomics in the future. And like I said, multi-omics is something we're all really keen to dig into methodologically. So we're working on a plan to make some more complex defined communities, maybe 20 to 50 human-derived strains, and then look at generating multi-omic data sets in culture and notobiotic models, really to provide a resource for all of us to establish and improve tools for multi-omic integration. If you have ideas for a resource or want to get involved with the benchmarking that IMPACT is doing, please get in touch. We would love to partner more with the community, and are anxious to hear your ideas. So how do you participate? You can scan this QR code and complete a short survey with your contact information, your interests. You can also get in touch with us. There's an email, phone number if you want to get in touch with us directly about this exercise. General inquiries can also go to the IMPACT email, and there's a website, of course. We'll be updating that in the coming weeks with all this information about the benchmarking. Thank you so much for your attention, and I look forward to working with you in the future.